Thank you very much for the introduction, Miguel. I think it was a very good summary about what I'm going to tell you today. So first, thanks a lot for inviting me. I really like this network. I think it's very interesting. I've been attending some of the seminars so far, and so it's really great to be able to give one as well and tell you a little bit about our work. So in my group, we are generally interested in microbial ecosystems. So not in one particular one, but in really trying to understand the general principles. So here I put up a couple of examples. So whether it's cheese microbes, soil microbes, and here I got a nice picture yesterday that Philip Engel sent to me to also demonstrate that I am in contact also with some people who are part of the SymbNET. And the idea in general is to look at these systems because we know that because microbes have very short generation times, their ecology and evolution tend to overlap quite a bit. And I think this makes them very interesting to study. But in addition, of course, they also are very useful in many aspects of our lives. So our overall goals are then to understand these systems in a way that we're able to predict what will happen to them in the future. So how they will interact with each other on ecological timescales, and then how they will co-evolve, with the ultimate goal of being able to control these evolving communities towards some desired outcome. So for example, a better tasting cheese, if you like. But rather than focusing on these systems, which we find are quite complex, instead we aim to be in the center of this diagram here, to have a few species that we can grow in the lab, so rather than some natural environment. And this allows us then to be able to work out all the interactions between different members of a community, even going down to a mechanistic level of what is exactly happening in these interactions. And just to mention, I know many of you work with the microbes associated to hosts, and for us, even this is maybe too complex, so we really try to simplify it one step away from that. So instead of a host, what we do, so these microbes actually come from this environment here. It's called the metalworking fluids. So these are liquids that circulate in pretty much any factory that does some machine cutting. So essentially they're used to cool down the machines, and these liquids circulate within the factory, and they're relatively toxic, so both to humans and to bacteria. And so they pollute the environment as well, and they need to be degraded. And so what our collaborators during my postdoc did, so they wrote this patent here where they discovered that if you isolate some bacteria that contaminates these liquids, you can then use them instead to degrade the liquids in order to remove the pollutants that are within them. And in particular, their patent mentions these 4 bacteria here that we are using now. So what these do is, if you put them into these metalworking fluids, which is what you see on the left here, when you put them in and leave them perhaps for a week, the pollution load is reduced. And so for us, this is interesting because it's a nice system where we can try to disentangle the interactions between the different species and really study its ecology. And it's nice because it's somewhere in between being a natural system, if you like, and being one that's manageable in the lab, because you can grow bacteria quite well in the lab. So what we've done then, since we've taken over the system, is to fluorescently label the different strains so that we can distinguish them from each other. We've characterized how they grow in this environment and how much they degraded, each alone and then together. And then, of course, we've sequenced and annotated their genomes, and we've started to measure the interactions. So today I'll be focusing on these interactions, and first telling you
I put the things that are on that table there as like things that
I think somebody needs to maybe mute their microphone.
Yes, I'm going to ask to everyone to mute ourselves, please.
Okay, thank you. Yeah, so the first part of the talk, we'll be talking about a study where we looked at the interactions between the species. And then secondly, we did an evolutionary study, and we tried to see how these interactions, whether they will change over time and in what way. So regarding the interactions, so this is a project by Philippe Picardi, who's just finished his PhD in the group. So what he did as a first experiment was to take each of these 4 species, first grow them alone, and then grow them in all possible pairwise combinations. And the idea then is to see, so if a second species is there, for example, how will this species AT grow compared to when it's alone? And so what he did then, so some technical details, but essentially what he does is he mixes this metalworking fluid. So it's basically an oil concentrate. He mixes it in water to make an emulsion, adds some trace metals and some salts. And then he puts constant cell numbers from each species together. So if we put 100 cells of AT here, we would also put 100 cells here, and then, for example, 100 cells of the other species. And then what he does is just measure the population sizes over a time scale of about 10 days. So this is some of the data. So if we look at this particular species here, AT, so this is how it grows over time. In green, you can see what happens when it's on its own. And then in black, you see how it grows in the presence of different partner species. So what you can see is when it's on its own, it tends to not survive so well. So it's a little bit stochastic, whether it dies or survives. And then with some partners, it actually grows relatively well. So if we do the same analysis then for the different species, we'll do this. You also get something strange there. Oh, here we go. So if you look at this also for the other species, we see that we have the other two species, MS and OA. So again, if you focus on the colors, the colored lines are the monocultures, and there you see that they die when they're alone, and they only survive in the presence of certain partners. And the only one that could grow alone in this case was CT. So we can quantify these interactions a bit more quantitatively. And basically, we measure the area under each of these growth curves and then do some statistics and then map that onto a network of interactions. So if you're curious, you can ask me at the end about exactly how this works. But we can translate it into this network of interactions where the arrows are either green for positive interaction or brown for negative interaction. And then we have the thickness of the line shows how strong the interaction is. And then we have stars for significances. And what you can appreciate immediately is that all the interactions that we measured were positive. So species, if they had any effect on another, it was that they grew better in the presence of another than they would alone. And so this was quite surprising to us, why we got so many positive interactions, because I had actually written a paper during my postdoc. So this was a review paper. And what the literature seemed to show is that different species are most likely to compete when we put them together. And so to explain this, we basically looked again at our growth curves. And what we saw is that there was a lot of death in this system. So we had 3 out of 4 species that were dying alone. So the environment is quite toxic. And the first hypothesis that we came up with and that we tested with a mathematical model that Björn Westman built was that perhaps it's this toxicity that actually explains the abundance of these positive interactions. So this was our hypothesis. And rather than showing you the mathematical model and sort of the equations, what I'll do is just show you a small animation. So this was actually animation made by Philippe to explain exactly what the model predicts. So imagine that you have an environment that just contains nutrients. And then you have, on the left, you have a monoculture, so just one species, this green one, that can grow quite a lot. But if it's in co-culture with another species that also eats the same nutrient, then of course it won't grow as much because they have to share the available resources. Now imagine that you put toxins into the same environment. So now the toxin doesn't allow the species to grow and perhaps even kills it. But then if you add now a second species in there, and if we imagine that this species degrades the toxin, in that case, what would happen accidentally, if you like, is that now both species are able to grow. And so if you compare this condition here to the one in the absence of the blue species, then you would say that the interaction is positive. But of course, underlying this is competition for the same nutrient. So what this basically tells us is even if there is competition for the same nutrient, there is a possibility that when you measure interactions that you would see positive effects between species. And so we wanted to test whether this model actually holds in our environment. And so what we decided to do was to take our initial environment, so that was metalworking fluids that we know contains both nutrients and toxic compounds, and then increase the nutrient concentration there. So we decided to add amino acids to this metalworking fluid in the hopes that it would make it a bit less toxic and that perhaps some species would be able to grow and maybe we would see more negative interactions. So the idea is here we have just a bigger apple, essentially. And indeed, what we found was if we measure the interactions here, that a few negative interactions popped up. And like we had hypothesized, so in this case, when we add the amino acids, we have one of these species, the green one, AT, becomes autonomous. So essentially, it's able to grow alone. So if you remember, in the original network, we only had one species that could grow alone. And here we have two that can grow alone. And as soon as they're able to grow alone, then we start to see competition between them. Whereas the other two species, MS and OA, were still not able to grow alone. So they still depend on the others in order to survive. And so we're not surprised to see basically that they receive all these positive interactions from others. And then as a last step, what we did was to remove the metalworking fluids completely. So we just put amino acids. And in this case, we expect that there's very little toxicity. And indeed, in accordance with our hypothesis, when it's even less toxic, then we get more negative interactions. And again, according to our partition of the species into a group that's dependent and a group that's autonomous, we see that it's the dependent ones that are the receivers of the positive interactions. And we see mostly competition between the species that can grow in this environment. So what this tells us is that interactions between species are not constant, first of all. So it doesn't make sense, for example, to take a sample from the environment and then try to measure the interactions between species and assume that those interactions will hold regardless of what comes their way. And we also predict that toxicity can promote facilitation between species. And here I've underlined facilitation just to distinguish it from cooperation, because cooperation, at least in evolutionary terminology, would suggest that that this interaction evolved for that purpose. Whereas here, what we think is that this detoxification accidentally helps other species. Okay, so this was quite nice, but the problem with these media is that they're all quite complex, and it's a little bit difficult to see if what we find is specific to this environment or it's something more general. So next, we are working on doing the same experiment, but in a more defined chemical environment. And so this is work that's done by Rita Di Martino, who's an experimentalist, so she's doing her PhD, and Aurore, who's a theoretician, and she's also building a model to help explain the data that Rita is generating. And so what Rita is doing is basically, instead of using the whole complex metalworking fluid, so we actually don't even know the chemical compounds that are in the metalworking fluid because they're covered by copyright. And so instead what she did was to take just one single compound that we know is commonly used to make metalworking fluids, and this is linoleic acid. And then she grew the species in linoleic acid either alone and together. And we also looked at different concentrations of linoleic acid. And what was interesting was that if you look at low concentration of linoleic acid, it's a nutrient for two of our species, AT and CT. But then if you increase the concentration, what you find is that it's actually toxic. It becomes toxic for AT, and then it's a nutrient for CT. So basically here, AT cannot grow alone anymore. And so what we would predict is that that would change the interaction compared to here. And indeed, when she measures the interactions, we find exactly this, that in the case where it's a nutrient for both, they compete. And then when it becomes toxic for one of the species and a nutrient for the other, then the interaction can switch to being positive to facilitation. So this is interesting because it shows that simply changing the concentration of one compound can already affect the interactions and even switch the sign of the interactions between two species. Okay, so that's the end of the first part of my talk. So looking at how these four species interact, what we find is that interactions are quite context dependent and that positive interactions may arise in harsh environments where species depend on others to survive. My second question and the second part of the talk is about the evolution of these interactions. So in terms of the experiments, what we decided to do, and this is again part of Philippe's work, was to put the four species together in this metalworking fluid, to grow these species, so in a batch culture for a week, and then to transfer part of the population, in this case 1%, into fresh medium, so to give them fresh nutrients and then to let them grow, etc. So to do this over 44 transfers, 44 weeks, which is approximately the scale of a year. And the question is, starting from these positive interactions here, what will we end up in the long term? And to have a control next to that, we also evolved the species, each one alone, to see what is the effect of the presence of the community on the evolution of the species. And this is actually something that's been done a few times before. So what we're doing is not completely new. But what is interesting is starting from a community where the interactions are positive. Because in many of the other communities, the interactions were competitive to start with. And so here we're asking, if you start with facilitation, do you, for example, end up with a cooperative community? Because you could imagine that if somebody helps you, then maybe you would evolve to be able to help them even more, because that would feed back onto you. Or you could imagine that this cooperation or this facilitation would break down and they would start to compete, etc. So in the literature, there are a few predictions like that, and we wanted to see which one holds in our case. Okay, so some of the data here. So here I'm only showing you 2 monocultures, and that's because the other 2 monocultures, the species died out quite quickly. So those were for MS and OA, which I showed you they really cannot survive on their own. And what we did was we had 5 parallel microcosms for each of these treatments, so the 2 monocultures and the co-culture. In this case here, so when AT was alone, in 3 of the microcosms it went extinct and it survived in 2. Now what I'm showing you here is the population size at the end of each week. So before we do the transfer, we see how many cells there are in our cultures. And what you see is that quite at the beginning, they almost died out. Again, because they couldn't grow very well, but in 2 of the microcosms, they quickly recovered and were able to keep a stable population over time. So again, imagine here every week we're diluting and then they grow back to the same population size, more or less. We see something similar in CT, but here we had all 5 microcosms that were able to survive. And then when we put all of the species together, we see that the 4 species can survive and end up with a relatively stable community. So the interesting thing is, despite them being different lineages, we got stabilization in almost all of the cases, except here we have 2 microcosms where MS went extinct. And in the other 3, even though there was a dip, eventually they recovered and they were able to end up in quite an even community towards the end. But what we're really interested in is to see how the interactions changed over the course of this evolution. So what we did was basically take isolates from the very end here, from transfer 44 at the end of the experiment. And then what Philippe did was to do all the monocultures and the pairwise interactions exactly like he did before for the ancestors. So just as a reminder, so this is the ancestral network of interactions. And then if we look, so we look at one particular microcosm where he isolated some of the strains. And this is the interaction network that we see now. So first, it's interesting that not that much has changed, in the sense that we still don't see any negative interactions, so it's still quite positive. But the positive interactions have mostly shifted towards these 2 species, MS and OA. And indeed, if we think of, again, about this labeling of whether species are autonomous or dependent, we see that, again, MS and OA are still dependent, so they still cannot grow without the presence of the others. But in this case, AT became independent or autonomous. So now it can grow on its own. So just as a reminder again, so this was the ancestral population. We only had 1 that could grow alone. And then if you remember, when we put amino acids in, we got a negative interaction here. So our prediction was actually, if one of the species becomes autonomous, then we would expect it to compete with the other. So the question is then, why here do we not see a negative interaction, even though both of these species can grow alone in this medium? And so we decided to focus on this species pair. And our hypothesis then is that the lack of negative interactions here is something that's specific to them being co-evolved, so them co-evolving together. So the nice thing here is that we don't just have the co-evolution treatment, but we also have the treatment where we evolve them alone. So the question is then, how about the 2 that evolved alone? Would they compete? And that would allow us to address this hypothesis and see whether it's really the co-evolution that is responsible for the lack of negative interactions here. So again, a reminder, so here we were measuring the area under the growth curves. Sorry, I haven't shown you this plot before, but essentially here we can see how much AT is growing on its own and how much it's growing in the presence of the other species. And here we care specifically about CT. So you see that it grows better with CT, and CT here is its baseline growth, and it grows better with AT. And that's why we had these plus interactions. Now we can do the same for each evolved alone, and this plot looks slightly different because we put both AT and CT together. So this is the monoculture, and the reason there are many dots is basically because we've taken many isolates from the last generation, and then we do technical replicates for each isolate, so that's why you have the little bars here. So we first grow them each alone, and then we grow them together in co-culture, and you see for many of the combinations, it seems that they grow worse when they're in co-culture compared to monoculture. So if we translate that into interactions, then they compete with each other. So they affect each other significantly negatively. Now, I showed you before the data from just one single microcosm, but then we looked at all the different microcosms of the AT and CT that had co-evolved in the same microcosm. And here we see no significant difference between growing them alone versus growing them together. So essentially, there is no significant interaction between these two species. So in summary, then what we went from is ancestrally, they interacted positively. If you evolve them alone, they evolved to compete. And if you evolve them together, they evolve to not interact with each other. So basically, this is what I'm summarizing here. So co-evolved species have reduced their positive interactions a bit against our expectations. Now, just very briefly, another interesting thing that we saw. So here on this plot, basically, you see the growth curve over 8 days of the ancestral bug, of the one that evolved alone, so that you see in the solid, and the one that evolved in the stripe lines in the community context. And what you see that if the species evolved in a community context, at least for this microcosm, they grew less well. So we then looked in all our microcosms to see whether this was general. And here you see the results. So we looked first at the total area under the curve, and we see that it's significantly lower for those that co-evolve. The growth rate of these species is also significantly lower for the ones that co-evolve. There was no significant difference in the maximum population size, but also if we measure how much of the pollutant they degrade, then we find that also the ones that evolved alone degrade a lot more than the ones that evolved in the community. So basically what this tells us is that we think that, that the ones that evolved alone, the ones that evolved together with the others in the community have been constrained in their evolution. So potentially they could have evolved to grow faster, they could have evolved to degrade more, as we saw in the ones that evolved alone, but they didn't, potentially because of the presence of others. So maybe just a little diagram to summarize what we think we're seeing here. So if you imagine the ancestors, we have 2 species here, and perhaps we have 2 nutrients, and some of the species have the potential to take up these nutrients, but they're not yet. Now, if you evolve them together, what we seem to see is that they end up occupying different niches, so that each one is taking up a different nutrient in order to avoid competing with each other. Whereas if you were to evolve each one alone, they would both evolve to consume all the possible nutrients in the environment that they could initially potentially consume. And so this would lead here to less competitive strains that are constrained in their evolution. And this is quite in line with things that have been seen in many other species. So this is called ecological character displacement. So this was a study that was done in Darwin's finches. So basically, you imagine if 2 species are on the same island, that they would evolve different beak sizes that don't overlap with each other. So each one eats a different seed size. Whereas if you were to evolve each bird, each finch on a separate island, then they would evolve to be able to eat all of the different seeds. That's what we think that we're seeing here. Now, I know many of the listeners are probably really interested in the genomics and are asking themselves what happened in terms of genomic changes in our experiment. So unfortunately, I won't be talking about that today, but just to tell you, so we did sequence the metagenomes of our bugs. So this is Mark, who is a postdoc in the group, and he sequenced together with Philippe samples, metagenomic samples coming from 4 different time points across the experiment. And then more recently, we've also taken clones from the last generation, extracted the DNA, and then sequenced that. So here we used PacBio, whereas with the metagenomics, we used Illumina. And at the moment, Eric, who's a new PhD student in the group, is analyzing that and comparing the 2 data sets. So this is still ongoing, and hopefully you'll maybe read about it once we have the paper put together. So just to summarize then, so the second question about the evolution of the interactions. So what we think we see is if species co-evolve, then they might constrain each other's adaptation and evolve to avoid each other's niches and avoid competition. So is this general? Then going back to thinking about all the systems that I talked about at the beginning. So of course, we don't know this. We've only studied this in our specific system, but we can at least make some predictions. So what we think is that when you put species together in the first place, so say you bring them into the lab and they meet for the first time, then their interactions will depend on the environment. And we think that the harder it is to survive in that environment, the more likely it is that they will interact positively. And then if you leave them to adapt to each other, we expect these interactions to weaken. So whether they were positive or negative from the beginning, we think that they will just become less strong over time. And then the next question that we're working on in the lab is community control. So thinking about whether we can actually direct these communities towards certain interactions that we would like. But of course, I don't have time to talk about that today. But maybe in a future opportunity. So just to finish off, I'd like to thank some people from the department who helped us a lot getting this work together. Kevin, who has discussed a lot of these thoughts with me during my postdoc. Two collaborators from Blaser Swiss Loop who make metalworking fluids. Of course, Giant Microbes for the nice pictures. The funding bodies that helped us to do all of this work. And of course, the people who did all of this work, so the really great members of the lab. And if you're interested, I'm currently looking for postdocs to do both theoretical and experimental work. So please do get in touch if you're interested. Okay, so I'll stop there. I hope I didn't take too long.
No, it was perfect. Thank you, Sara, for the great talk.
Thank you.
Okay, so now we open the question session. Is everyone want to ask something to Sara? Sure, Karina, you can unmute and ask.
Hi, Sara.
Hi, Karina.
Nice to see you. Thank you very much. Very nice presentation. So first, if I understood, just as your last message is that if they are co-evolving, you expect them to be evolving towards less competitiveness, and they will be more constrained, but finding different niches, and so there would be less strong interactions. Is that correct?
Yes, that is what we predict. And there have been a few other studies that have shown this also in competitive networks. So if some species start with competition, that these competitive interactions tend to weaken over time.
Mm-hmm. So and that would be like, for example, in a microbiota type of situation where you expect to have a few interactions.
Yeah, I think it also depends a little bit. Sorry.
Weak interactions, right?
Yes, we would expect weaker interactions, but it really depends on them coexisting in the same environment for a long amount of time. So what's maybe specific to our environment is that we don't get any migration. So it's always the same for species that can adapt to each other. And I suppose in a gut or in some other environments, you would have more incoming species. And perhaps every time a species comes in, then there might be strong interactions. But if they maintain coexistence over time, then one might expect the interactions to weaken.
Okay. And regarding the first part, I was wondering because you said that there was a lot of killing due to the toxicity. I was wondering if you considered in your model the possibility that they would be growing better because they're eating the nutrients from the others when they lyse.
Okay, so when they lyse, they don't. So that's something we try to test for. So we killed our cells. Basically, we tried to lyse them and then feed them to each other. And if anything, there was a slight negative effect. So it might be that there's also toxic compounds inside the cells that then get released. But yeah, we didn't find that. We do find sometimes a little bit of cross-feeding in simpler environments. So that might play a small role here. But we think that the dominant effect is because of detoxification.
Thanks.
Okay, you also have a question for Anasus Taipas. You can unmute and ask. Go.
Sure.
Yeah.
Hi, Sara. Very nice, very clear. I was wondering on the second part about the constraint in evolution when you're part of the community versus alone. I mean, evolution will always depend on the number of generations you have, and same days would not mean the same number of generations when you're alone or when you're part of the community. So how much do you think this is confounding things?
So I actually don't think the generation numbers are that different in our case. I think we'd have to quantify it. But just looking at sort of the population sizes that we get, they're not that different between the two conditions. So if you focus on the green line here, for example, and I think it was the same for the blue, but it's true, I would have to calculate that. I mean, what might be a difference between the two conditions is the bottleneck at the beginning is quite strong. So that might be something that would lead to differences between the two. It might be accumulating more mutations at the beginning. But again, I mean, I hesitate to say too much because we're currently doing that analysis to try and figure out what happened here. Yes.
Okay, I think you have also a question from Thomas Bosch.
Yeah, just a second. Okay, thank you, Sara. Fantastic work. Yeah, very inspiring.
Thank you.
My question is pretty simple and naive, but this is your modeling. This is all, in my naive interpretation, kind of biofilm communities. So they are living somewhere and with themselves, and then you find and you discover the rules how to do that. Could you speculate if the same mechanisms also hold on for a microbial community living in a host tissue?
Yeah, so I suppose if you're living on host tissue, I think if I understand the host environment correctly, that there's also, I'm not sure how many species are able to survive on their own in a host environment. So my understanding is, for example, that there are bile acids within the guts that might be difficult to survive. And so perhaps we could have some species that are able to deal with these bile acids, for example, by secreting enzymes, and that might facilitate the survival of other species. So this is speculation, right? I don't work on the gut, but it would be interesting to see.
Yeah, I just wonder, I mean, people try to understand how the gut is complicated. And by the way, bile acid can even be a nutrient for some of the bacteria, at least some components of it. But on the skin, for example, so people do not understand at all how the community interacted. The complication is that the host factor may also positively, the host, because of coevolution, may also positively interfere with that community. And so I'm wondering how much more complexity do we need? I mean, you could consider the host as an environment or a different environmental factor, but it's probably a little more complicated. So I just wonder how easy one can transfer your findings to for example, the simplest thing is skin microbiome or in your oral cavity. There is not much. It's just on a mucus layer bacteria, but they interact with the host cells and the host cells interact with them.
Yeah. So I think there's a few levels. So one could see the host simply as an environment, and then perhaps it's not a constant environment like what we're looking at, but it's something that's fluctuating. So for example, there's food intake that's coming in and then maybe nothing, or there's the immune system is attacking the cells or not. So I guess there's lots of fluctuations. But I think what's also interesting is to think a little bit about this question of control, because presumably a host also wants to control its community. And so if you imagine sort of an evolving host might sort of have an interest in evolving towards creating more positive interactions between the species that it harbors if they have a certain function, so then maybe they could be more synergistic, etc. So I think an interesting question there is to what extent can a host control its communities? And I think there are one or two papers that sort of discuss this idea. But for sure, I mean, we start simply without a host, and then the question is if you were to add a host, what does that do?
Sure. Thanks.
Thank you.
Now we have a question from Maria. Go ahead, Maria.
Thank you. Hi, Sara. Very nice talk. Thank you very much. I have maybe a quick question regarding also this the statements that you have on the slide, right? You say that interactions depend on the environment, and as they evolve, they may weaken. So my question is whether how the interactions evolve would also depend on the environment or not, because in your model, like your species have the possibility to find niches, right? But what in the environment where there is no possibility for niches, or where there is like constant change of nutrient, do you think there might be other types of interactions evolving, and are you planning to test them experimentally somehow?
Yeah, that's a good point. Yeah, because the environment that we're looking at does have a lot of niches. So I think it's chemically quite complex. So presumably they can use that to sort of, yeah, each focus on one part of these niches. And it's true that if we imagine, for example, we've had a single substrate, that would look different. However, what we've done so far, so we have tried, so I mentioned briefly this linoleic acid project, and we have done a few transfers, at least in linoleic acid, and it seems like there's surprising coexistence in that system. And so I think what we're finding, and also other labs are finding, is that there's more and more cross-feeding that allows for more niches to be generated, basically, that niches get constructed, even if you start in a very simple environment. Also, chemical changes occur over time as molecules oxidize and so on. So I think niche availability is, and my guess would be that it's not very constrained.
Thank you.
Okay, unfortunately, we don't have more time for questions now, but you are encouraged to contact SymbNET and to schedule any time to talk with Sara if anyone have more questions, okay? At the end of the other talk. Thank you very much once again, Sara,
Thanks for your question.
for your talk.